All right, welcome back. Antigens and antibodies. So recall that antibodies are globular proteins that we call immunoglobulins. And you can see in this image why they would be called globulins, right? So immuno because they have a function with the immune system, and globulins because they look like globs until you magnify this further to see the actual structure. And you can kind of see even in this resolution, you can see this kind of Y shape, and we will be looking at that Y shape. Let me go ahead and bring that up. So this is the Y shape, and so further study has revealed to us what's going on at the molecular level. Okay, so let's talk a little bit more about antibodies and antigen, and then we'll start diving into more details. So recall that plasma B cells are the ones that they make and secrete the antibodies into your serum. And then of course you have the memory B cells with that same antibody, but it's embedded as receptors, and they can immediately become plasma B cells if they encounter that antigen again. So antibodies will bind to antigen, and we're going to look at exactly how they do that. So antigen is simply any substance that causes the immune system to produce antibodies. So any substance could be, any part of a bacteria, any part of a virus, that stimulates an immune response that leads to plasma B cells, right? So there's a huge diversity, and I already started talking to you about this in the last lecture. There's 100 million trillion antibodies in your body. That's an incredible number. Take a moment, take that number in. So why do we have so many antibodies in our body? Every time you encounter an antigen and make antibodies against it, those become circulating in your blood. So we can attack a few million different antigens. And of course, you wouldn't have just one antibody per antigen, right? You're going to have lots of antibodies per antigen. We have the ability to detect about a few million different types of antigens, and this is due to that variety when we're producing our B cells, when we're producing our T cells, when they're gaining their receptors. Remember, we talked about there's a portion called the variable region. It's the top part that binds to antigen. And our body simply just makes a bunch of random changes to that variable region, just making as many different variations as possible. And then, of course, we have to check that none of those will be harmful. And so clonal deletion, right? So our body has made clones essentially with just very, very slight differences in the variable region, just making every possible combination, right? And then we have to make sure that none of those will bind to our own cells. If it does, we kill it, we get rid of it, it's eliminated, it will not be allowed to leave and become part of lymph, right? But if it passes, then it will go ahead and be allowed to mature and go into the lymph. So remember, we talked about different functions of antibodies. One role that they do is to enhance phagocytosis, and that's called opsonization. This is by coating pathogens with antibodies, making it easier for phagocytes to grab on. There's agglutination, which is clumping, whether it might be clumping up bacteria or bacterial parts like LPS, or clumping together viruses, right? So agglutination just is to clump. Of course, eventually that will be also have to get taken up by phagocytes. And then we also learned that antibodies have the ability to activate complement. And earlier in the first lecture, we learned that antibodies can also trigger what's called antibody-dependent cell-mediated cytotoxicity. All right, so how exactly do antigens and antibodies interact? So that top region of the antibody is called the variable region. It consists of two arms, one that's called the short and one that's called the long. And notice that, so the short chain has two regions, the top part being part of the variable region and the bottom part being part of the constant region. So all antibodies will have the exact same blue regions. Part will be in the short chain and part will be in the heavy or long chain. Notice that they're bound together through very strong covalent bonds. And there's two chains. The top part of each chain makes up the variable region. And very specifically, the part of the variable region that does the actual binding to the antigen is called the paratope. So the paratope is on the variable region of the antibody. The epitope is on the antigen, and it's the part of the antigen that binds to the antibody. Okay, so paratope and epitope are the most specific terms that you can use for binding of antibody to antigen. Okay, and that antigen could be attached to the microbe, or it might have become detached from the microbe. It doesn't matter. But our antibodies are tiny, and they're only going to bind to a tiny region of an entire antigen. Here, this is looking like a white blood cell, whether it be well, it looks like it's a B cell, but we don't know, it's not labeled. But this has a receptor that is an antibody, in fact, but it's bound into the membrane and it's binding to antigen, and then we see it up more closely and more closely here. So the antigen's epitope, the antigen's epitope binds to the antibody's paratope. That's correct. And the paratope sometimes is also referred to as the antigen binding site, because it's exactly what it is. It's the site where antigen binds. Okay? So this is showing you the same thing again. However, now you're able to add another layer in that on a microbe, you know there's going to be all sorts of different antigens. And this image only shows 3, but there's definitely more than 3 types of antigen on a microbe. And it can be inside the microbe, and it can be on the surface of the microbe, right? It could be flagella. Any molecule essentially can act as an antigen. So this image shows 3. So you would have one type of antibody labeled A that could bind to this antigen. And then you would have to have a whole different antibody, right? A different variable region that could bind to these dome-shaped antigens, right? So this is a different antibody. And then you would need a third antibody if you wanted to tackle these square antigens. And so on and so forth. So when you get an infection, say with a bacterial infection, you could potentially be making a dozen different antibodies because you've been exposed to different parts of that bacteria. And that is great because now you have even more protection, right? You're more likely to detect that bacterial cell in the future and prevent infection. Okay. So antigens evoke an immune response, and that will signal the body to produce antibodies, so plasma B cells and memory B cells. But it also, antigens, once they're presented to T cells, can initiate cell-mediated immunity as well. So don't forget about your T cells and cell-mediated immunity. The only difference is B cells can actually grab antigens out of solution, right? Free antigens. Whereas T cells have to have antigen presented to them. So there's a difference there. But typically, if antigens are present, you're going to stimulate both branches of your adaptive immune system. And remember, antigens can be anything: cell receptors, viral spikes, DNA, RNA, because the DNA and RNA of pathogens will be different than our own nucleic acid. And we can distinguish between them. Carbohydrates that are not found in humans, such as peptidoglycan or, chitin, if it's a fungi. Proteins not found in humans. I don't have any specific examples of a protein offhand, but there are going to be different types of proteins that are common to bacteria or fungi and are not found in humans that can be good targets. So essentially, any microbial molecule that's not found in a human and stimulates the immune system to make antibodies. Okay, so we started talking about this light chain, heavy chain, like I guess you could say the anatomy of an antibody. So overall, you have 4 polypeptides: short chain, long chain, short chain. So those are the 4 polypeptides, right? 1, 2, 3, 4. And we call them light and heavy because the short ones are going to be lighter than the heavy ones. Interestingly, the 2 heavy chains are connected together, and then the light chains are on the outside. Okay, so that's the structure, and it makes kind of that Y shape, which is why when people draw antibodies, they draw them as a Y. Okay, the variable region is found on the tops of each light and heavy chain. And at the very end of each of those, you're going to have an NH4 group. So the NH4 end is on top with the variable regions. And then the other ends, which are constant, meaning every single antibody will have the same structure for the rest of it. And those will all end with a COOH group, right? So constant region means all antibodies have that same amino acid sequence, right? And antibodies will only differ from each other at the top, which is the variable region. All right. Strong disulfide bonds link the chains together. So there's 5 classes or categories of antibody. We just name them A, D, E, G, and M. And they are, we say Ig for immunoglobulin. Types D, E, and G are monomers, the classic Y shape, whereas IgA is a dimer, 2 antibodies held together in this shape. And then we have a pentamer where you have 5, 1, 2, 3, 4, 5 of the antibodies hooked or connected together. And we'll see pentamers are really great for agglutination, right? Dimers can do agglutination too, but the pentamers are really the main agglutination. Monomers are, we'll see a lot with like neutralization, activation of complement, opsonization, right? Whereas The other two are more useful for agglutination. Now we're going to look even more specifically, to where each of these are found in the body and so forth. Okay. Okay, so before we look at each individual, something to keep in mind is this idea of affinity, which is the like the attractiveness of two objects to bind to each other. So how likely is the antibody to bind to the antigen? If it has a strong or high affinity, you expect both of the binding sites to be bound to antigen. If it had a low affinity, then you would maybe just have one instead of both. Okay. Now, avidity is the sum, and when you have something like this monomer, and also think about how many, hundreds of thousands of antibodies you would actually have, then you want to think about the cumulative effect, the sum of all of the affinities. And so maybe you have a medium to low affinity on one pentamer, but when you have, dozens of, 2, 4, 6, 8, 10, you have 10 binding sites on just one. Essentially, you would consider this to be one antibody because they're all connected together. It's one IgM antibody. So even if those have medium to low affinity, you add so many binding sites up together and it can have a large impact. Kind of like how we talked about with hydrogen bonds and the DNA molecule. Hydrogen bonds very, very weak, but when you have millions of them, it's actually strong. And that's why it takes, high temperatures to separate the strands of DNA to break all those hydrogen bonds. Now, there's actually 4 different types of weak bonds that can form. So I talked about hydrogen bonds. We also have something called van der Waals, which we're not going to get into, but it's a little more abstract and even weaker than hydrogen bonds. Then we have ionic bonds, which are generally stronger than hydrogen bonds. And you have hydrophobic interactions, and I can't really put that on a scale as to how strong that is, but it's called an interaction, so it's not going to be very strong. Overall, these are all weak, right? Covalent would be very strong, right? So don't mix those up. All right, so IgM, there's not usually very much IgM circulating in your serum when you're healthy, okay? This is the pentamer, the ring, right? It can fix or activate complement, but its strength is agglutination, as I mentioned earlier. Also, this tends to be the very first antibody produced in response to infection. So when someone is first experiencing infection, you're going to, once it's been enough time for them to be produced, 7 to 10 days post-exposure, then you're going to see a very high level of IgM antibodies in their serum. Okay, so this is the first responder, the first antibody made in response to infection, and its primary job is to agglutinate microbes. Let's get all of those microbes together in a clump so they can stop infecting you. Okay, you can find it in blood, lymph, and even on B cell surfaces. Okay, then we have the IgG antibodies, and these will take over. Right, IgM is first in response to infection, and then IgG will end up taking over and be made to a much larger amount. And we'll see this in a graph in a moment. So it's a monomer, as you can see, it's just one unit. Okay, most of the antibodies in your serum will be monomers, 80%. Okay, and specifically IgG. these are good at activating complement. They're really good at enhancing phagocytosis, so opsonization, right? They're good at neutralizing toxins. And also, since these are so tiny, they can cross the placenta and protect the fetus. And those last half-life of 23 days. So that's pretty long, long time that they can circulate. And you'll find them pretty much everywhere: blood, lymph, your intestines. Okay, and again, it can cross the placenta. So lots of very important functions here. IgA, on the other hand, is pretty much only found in your mucosa, right? So mucosal protection, find it in your mucus, saliva, tears, and even in your breast milk. Short half-life, 6 days, okay? This makes up a rather large amount of serum antibodies when you consider that their main job is to be in the mucosa. And it's a dimer, 2 antibodies bound together. And last but not least are IgE antibodies, which are also monomers, but these you don't really find in the serum very much. They're mostly free in the blood when you're making them, but their job is to coat parasitic worms and start the antibody-dependent cell-mediated cytotoxicity, okay? So these have a super short half-life of just 2 days. You don't want these circulating and causing tissue damage, right? You want them only when you need them and exactly where you need them, and only for as long as you need them, right? Because you're going to cause all those white blood cells to throw all their toxic substances to kill the worm. If you don't want it to be doing that to your own tissue, right? Okay. These are also tend to be the antibodies that we find when people are having allergic reactions. Not every allergic reaction is due to an antibody, but we'll look at that briefly at the end. Okay. So you first get exposed to, let's say, a virus. So we have a lot of viral antigen in our serum during the peak of that infection. This immediately triggered an immune response, but remember, it takes 7 to 10 days to make antibodies. You will see an increase in your IgA, assuming that this virus is something like a respiratory or gastrointestinal infection. So it's first stimulated your mucosa, and then you get your IgM, those pentamers. And then, as I told you, IgG will eventually take over, and it gets made to a much higher level than any of the other antibodies. Remember, IgG is a monomer, IgM is the pentamer. So looks like I wrote the wrong letter here. Please fix this. It should say IgG, right? See how the blue IgG takes over. Yeah, I apologize. I didn't catch that. Please fix this. Okay. Red. IgG. There we go. All right. I just want to go back to pointer. There we go. Okay, so antibody titer. Titer is the amount. So antibody titer is going to mean the amount of antibody in the serum or sample. Primary response is the response after your first contact with that particular antigen. And you, if you produce, a memory response, typically you would, right? There's a few exceptions, but most of the time you do stimulate a full immune response and get the memory. Then, assuming you have that memory, then if you ever come in contact with that antigen again, you'll have a secondary response, which is much quicker. much larger volume and last longer. So let's take a peek at that. So you have your initial exposure, primary, you get your IgM, and then you get your IgG, right? Just like we talked about. But sometime in the future, far in the future, doesn't matter, sometime in the future, you come encounter with that antigen again. Now your memory B cells and T cells are going to instantly respond, and look at that level of IgG. You get more than twice the amount and so quickly. Your IgM still responds and it looks pretty much the same as the first time. Nothing impressive. It's really the IgG that is impressive. And so it's the IgG antibodies that are primarily that are giving you your lifelong immunity. Okay, now what this doesn't show is it doesn't show you your T cell cell-mediated branch. This is just focusing on antibodies. So this blue line here is the IgG antibody. Okay. So if you wanted to compare those in another way, so this is one way of looking at it. Another way of looking at it, so the plasma antibody concentration over the day, a week after exposure. And so you have your primary response, and it can get quite high with the amount of antibody. It took a while to get to that peak level. Now, when you have a secondary exposure, you immediately secrete an insane amount of antibodies. Look how this is so far above anything that you did the first time. And it stays, even though the levels do start to drop off a little, it's still higher than the highest point of your primary. So this is why secondary response with those memory B cells is so cool and so effective. This is why vaccines are so powerful, because you don't have to wait for the primary response to happen from having infection. The vaccine is giving you your primary response. And that way, if you ever come in contact with that pathogen in real life, you immediately have this secondary response and you're protected. Okay, so you can naturally acquire your immunity, and it can be active, and this would be the result of an infection. You can naturally acquire antibodies, and it could be a passive acquisition. This would be things like transplacental during pregnancy, or after birth during breastfeeding, right? That's totally natural for mother to pass antibodies onto her baby. But it's passive because the baby did not do anything. The baby's immune system didn't do anything. It just gained those antibodies. And once those antibodies are used up or wear out, they're gone. And the baby's not able to make those unless the baby goes through the process of making its own. Okay. Then you can have artificially acquired active immunity, which is vaccination, right? It's artificial because you're being given a shot of something that's not going to cause an infection, but will stimulate the immune system. And so it's active because your immune system has to react to the antigen and make antibodies and memory T cells. Then you could have, the last one is not very common, artificially acquired passive immunity. This would be like antibody therapy. Someone's in the hospital, they have a life-threatening infection, and they're given antibodies to try to help them fight the infection. And this is becoming more and more popular. We saw it kind of experimentally used during Ebola outbreaks. And I think we've seen it with COVID a little, although I haven't heard it talked about as much as I expected. This is a direction I think we're going to see more and more antibody therapy when someone's in critical care. I think we'll see this. The thing is, we have to have the ability to make the antibodies and have them available. So we don't just have, every type of disease. Like, we don't already have antibodies ready to give to people. But still, I think we'll see more and more, especially like more common diseases that we don't have a vaccine or a treatment for. Yeah. Okay, passive immunity, as I mentioned, persons receiving antibodies. And so once they're used up, once they're used up, they're gone. And so the monoclonal antibody therapy is what I was just talking about, with a patient in the hospital receiving antibodies to help them fight their infection. And so that would be both passive and artificial. Whereas a mother passing antibodies to her baby is natural, but it is still passive. Once they're used up, they're gone. So this type of immunity is temporary. Since the antibodies will be used up or worn out, they will disappear, right? And then the recipient will be out and will not have protection anymore. Active immunity, your body is making the antigens and typically will lead to a memory response. You can get this from natural exposure, you get sick, or artificial, you get vaccinated. So let's talk about vaccines. Vaccines are targeted against diseases that have a high mortality rate. Lots of people die when they get when this disease is going around, or it might not kill a lot of people, but it might disable people. So survivors will have lifelong complications that are devastating. So it can be both, it could be either or, but that's how we choose what to try to make a vaccine against. Of course, there's lots of diseases that we've tried to make vaccines against and have failed. So we would have more vaccines than we currently have. It's not always doable. So I want to dispel some, there's a lot of false information and fears around vaccines, and this typically is for people who are not educated in the biology, right? I don't want to say they're not educated at all, but they don't know about molecular level biology. So once you understand how DNA works, and once you understand how antibodies and antigens work, then you really shouldn't have any doubt or fear about vaccines. So people fear that they will actually get the disease, which for almost all of the vaccines is not even possible. The exception is the live attenuated vaccine. So we currently have a couple of vaccines that are a weakened form of the pathogen, and we call those live attenuated vaccines. We only do this when we have no other choice. Okay. This does, however, give the best immune response, and so these are the most effective vaccines, but they do come with that slight rare risk of someone's incredibly immune deficient, for example. There's a slight chance that the pathogen will still be able to cause an infection, but it's very rare. Okay, what is common that people mistake as getting sick is because their immune system gets busy to make these antibodies, they can have a slight fever, tiredness, achiness, and they think that they are sick. Okay. Remember, vaccines are protecting you from, life-threatening diseases. So if you were to actually get sick with the actual pathogen, you would be like very ill and experiencing very, very extreme symptoms, not just a mild reaction. Okay. So every other type of vaccine besides the live attenuated carries zero risk of infection. And that's the vast majority of our vaccines. We're going to go through the different categories of vaccines and I'll show you which ones are which. It is rare, but sometimes people can have allergic reactions to one or more of the ingredients in the vaccine. Each vaccine is different. So you would have to identify what you're allergic to. There can be some overlap where there's like a couple of different vaccines that use the same, let's say, preservative, and then you wouldn't be able to get those. But again, most people won't have any reaction whatsoever. But I did want to talk about it. Even though it's rare, it is possible to have an allergic reaction to one of the ingredients. If you're curious, you can go to the CDC and you can look at the full list of ingredients in every single vaccine. Now, another common misconception, which I think is possibly not at the forefront of vaccines anymore, but for quite some time, we've had groups of people refusing to vaccinate their children because they're afraid that it's going to cause autism or some other learning disability. Autism is a genetic disease. Vaccines there's no chance, there's no way for vaccines to alter your genes. So no genetic disease can be caused by a vaccine. And you guys learned in our lab a little bit about what it takes to change, to insert or remove genes. You have to have specific sequences so you can target the DNA. You have to have enzyme to cut the DNA, right? There's just all sorts of steps involved in order to target DNA and to change it. And a vaccine simply doesn't have any of that involved. Vaccines just have antigen, and there's no way it can do anything to your DNA. Okay. So what's the goal of a vaccine? It's to get enough people immunized that that disease stops being propagated in the population. So we call this herd immunity, and I'll show you what that means. So when a majority of the population becomes immune, they're vaccinated. The few people who don't get vaccinated, maybe they're too young, maybe they're too ill. There's some reason they can't have the vaccine. And it's okay because everyone else has it. And so then we're able to stop the disease from becoming an epidemic, right? Again, vaccines provide artificial immunity, but it is active and very powerful. So here we have a group on the left. The blue people are not immunized, but they're healthy. Then you have a few people who are red, and these are also not immunized, but they are sick. So they're carriers of the disease, not just carriers, but sick with symptoms and contagious, and potentially spreading this disease to everyone they come in contact with. And so you can see in the instance that nobody is immunized and a couple of sick people arise, they can spread that very rapidly in this population, and most people will end up sick. Okay. Then you have a scenario where you start to have a few people who are immunized and they're healthy and they're protected. And they might protect, you know, 1 or 2 people that they're close to. But for the most part, you're still going to have the majority of the population get sick because they're not protected. So this is why, for instance, with our current vaccine and COVID, it's been a slow process, and we're still in the process of reaching herd immunity because we started out with the oldest group of the population, 75 and over, and then went down to 60 and over, right? And gradually more and more people slowly are getting vaccinated, but there's still the majority of the population that are not vaccinated and a chunk of people who are contagious. And so you still have the issue of the majority of the population being sick. So this is where we're at right now with COVID. There's a few people not sick, right? It decreased a little bit, but not much. What we're trying to do is get herd immunity, where the vast majority of the population has been vaccinated. All the yellow people are vaccinated. Blue, not vaccinated. Red, sick. Okay. And now there simply are not enough people to spread to, right? This person, they're coming in contact with these people, and those people are all immunized, so they don't spread the disease to anyone because everyone's immunized. So this blue person and this blue person, they're safe because everyone around them is vaccinated. So this is the idea of herd immunity. You get the majority of people immunized, the disease will not be able to spread in the community. Now, this is an extreme example. It's still possible for a few people to become ill. In fact, see, you went from 2 to 3 in this example, but you're not going to have a massive outbreak like you have here. So that's the idea of herd immunity. Get your vaccine if you have not been vaccinated yet. Check your other vaccinations. Make sure you've received all doses of your hepatitis vaccines and everything else. Tetanus is only, I think, once every 10 years. So you're probably current on that, but double check. Make sure you have good records of your immunizations and that you're on top of it. If you did not finish a series, you have to start over. So again, get vaccinated for COVID, then look into your vaccinations and catch up on anything that you're behind on. Okay, so the live attenuated vaccines are the only ones that carry a slight risk of infection. It's very slight. Okay. And this is a weak form of the virus or bacteria that typically will not be able to cause any disease, but it can very slowly replicate, and that's why you can get such a strong immune response. So it produces a very strong memory response, both cell-mediated and humoral, right? Humoral antibodies. Right now, the vaccines that are live attenuated that everyone in the United States should have, the MMR, mumps, measles, rubella, the varicella zoster, chickenpox, shingles, rotavirus. Rotavirus is optional, but it is recommended for children because this causes extreme diarrhea, and children are very, very susceptible to dehydration, which can cause death. So we try to give the rotavirus vaccine to prevent that dehydration. Okay. So rotavirus is a gastrointestinal, right, virus that causes extreme diarrhea in children. But it is technically optional. Then there are sometimes some influenza vaccines are live attenuated, but not all of them. There's so many different influenza vaccines. that they'll fall under all of the categories. Yellow fever would only be given if you're traveling somewhere where yellow fever is an issue. Polio, same thing, only if you're going to areas where you could get exposed to it. And then smallpox is still considered eradicated. So no one really receives smallpox vaccines, except for maybe some military people. But yeah, the smallpox is not really given it, not even if you're traveling, because shouldn't matter where you go, you should never be exposed to smallpox. It's been wiped off of all populations. Okay, so the first two are in bold. I would like you to remember that the MMR, mumps, measles, rubella, and the varicella zoster are the live attenuated vaccines. They provide the best immune response, the best protection. Okay. Now, a lot of inactivated, these are completely dead. They're not able to replicate at all. Not even immune compromised. Like there's zero risk of infection for all people. Because they're not alive, it's always going to require multiple doses to get a strong enough immune response. Okay. So immune response mostly will be humoral. You're not really triggering the cell-mediated portion of the immune system. And the amount of antibodies that you're producing will diminish over time, which is why periodic booster doses are needed to get that level of antibodies back up in your blood so that you are protected. Sometimes this is the entire pathogen dead, and other times it's fractions of the pathogen. And in a few cases, it's the toxins that the pathogen produces. So whole cell or whole virus, we have hepatitis A vaccine, which is the whole dead virus, the rabies vaccine. And remember that rabies is caused by the Lassa virus. We no longer have any bacterial whole cell or whole virus. Well, it's bacteria. We don't have any whole cell bacteria inactivated vaccines anymore. We used to, we've replaced them. We still have fractional ones, like some of the influenza vaccines, and your hepatitis B. Okay, hepatitis B. We HPV, human papillomavirus, which is now being given to young women. I believe once you're, gosh, I don't know what the cutoff is. It's either age 25 or age 30. I can't recall. But once you're over, let's just say over 25, they're not giving the HPV vaccine to you because they'll assume you already have it. So that's how common HPV is. It's everyone's going to have it, everyone who's sexually active. And so it's assumed by the time you're 25, you'll have been exposed to it and you'll have it. So the idea with the HPV vaccine is that we're giving it to girls when they're young enough, before they're sexually active, right? So we might be targeting, like 10-year-old, 10 to 12, is where the big push should be, but catch them before they're sexually active. Why are we doing this? Because there's a high risk of cancer. This is HPV is associated with inducing cervical cancer. Okay, cervical cancer. We also have an anthrax vaccine, which is not typically given, but if you were doing research, for instance, then maybe you would get the anthrax vaccine at that point. Diphtheria and tetanus, we actually use they make toxins, and that's what, tetanus is a neurological toxin, and diphtheria can injure organs. And so And that's how they end up causing death. So we use those toxins to stimulate antibodies to be made, but they're inactivated, so there's zero risk that it's going to cause any problems. Okay, so again, the things in bold, the specific vaccines in bold, I would like you to remember, and all of the general information. Okay, types of vaccines continued. Okay, pure polysaccharide we try not to do because it's kind of the weakest of all. This is going to give you that T-independent response where you get immediate small amount of antibodies. You're not going to have any memory response, so it's very short-lived. So most of these vaccines, we've figured out how to make them in the different categories, and so we don't give the pure polysaccharide vaccines anymore, with the exception of Salmonella typhi. If you're traveling somewhere where Salmonella typhi is a possibility, it's prevalent in that community, it's likely that you would get this, then you would be given a vaccine before your trip. So anytime you're traveling somewhere that doesn't have a very good water treatment system, you should definitely get this, right? But it's only going to last long enough for your trip, essentially. So every time you travel, you would need to get another vaccine. Okay. So what we've done is we've taken these polysaccharides and we've conjugated them to a protein so that they will actually have a increased immune response. So Hib, pneumococcal, and meningococcal, they used to be pure polysaccharide vaccines that we've now modified, conjugated to give an enhanced immune response. So what is Hib? Do you remember? It's a bacteria, Haemophilus influenzae. And I know you see influenza and you want it to be a virus, but it's not. It's Haemophilus influenzae type B. This infects the epiglottis and it can swell up and completely block your airway, and you can't breathe and you suffocate. And this, lots of babies have died from this. And so we give this vaccine very early to prevent infant death. Okay. I think you don't really get this vaccine again. I don't believe. I think it's only infants that we're concerned about. Whereas pneumococcal and meningoccal, anytime you're going to travel, anytime you're going to live in a dormitory or any other, large population type quarters, so dorms, military, prisons, nursing homes, all of those people living in such close contact with one another, that's when you really need these vaccines for type of, right, pneumococcal and meningoccal can both cause, you can get life-threatening pneumonia. It can even cross the central nervous system, cause meningitis, encephalitis. Meningoccal meningitis is in the name, right? Okay. Then we have our recombinant DNA technology, which we started to make a lot of vaccines this way. You insert the gene for a viral or bacterial, doesn't matter, but you provide the gene for the antigen that you're interested in, and then the yeast produce lots and lots of that antigen, you purify it, and there you go, you have your vaccine. So it's a way to provide purified antigen. So our hepatitis B vaccine is now, this is the one most commonly given, although we did see it back here, fractional, right? So some hepatitis B vaccines are from growing that virus antigen in cell culture and purifying it. This takes a lot more work. Essentially, once you have modified yeast, it'll be much easier to extract and purify it that way. So we do have 2 different hepatitis B vaccines that you could possibly get. HPV, I talked about earlier as well, human papillomavirus. And so, again, 2 forms you can have directly from the virus or from recombinant DNA technology. And then, of course, we have certain influenza vaccines that will be made this way as well. So probably, because influenza pops up like in every category, probably not going to ask you about that. So I guess I didn't really need to capitalize or make it bold. All right. Now there's a group. This one is probably the most confusing group, where it falls under recombinant DNA technology, but it's a live attenuated vaccine. It's very weird. So this is where we have genetically modified the salmonella so that it cannot cause illness. So earlier we looked at Salmonella typhi being a purified polysaccharide, and it's not a very good vaccine. It's not very effective. It doesn't last very long. Now we learned how to use some DNA technology to modify the Salmonella typhi so that it's not able to cause illness, but it can cause a much more effective immune response. So if it's possible, if you're traveling and you need the Salmonella typhi vaccine, this would be the one to get. This will be much more effective at preventing the disease. Remember, Salmonella typhi causes typhoid fever. It's not the same as the gastrointestinal salmonella that we're used to talking about. So when you're being careful with your chicken and your eggs, poultry, so that you don't get diarrhea, that's great. That's not the same as Salmonella typhi, which can cause a life-threatening, very severe disease called typhoid fever. Okay. You can go back and look at your survey of bacteria slides if you want to refresh your memory about Salmonella typhi. All right. And now the newest category of vaccines, the mRNA vaccines. So, Pfizer and Moderna COVID-19 vaccines are what we call mRNA vaccines. Now, there's been a lot of confusion because we had not been giving mRNA vaccines before. But this research started quite some time ago. We have been successful in making mRNA vaccines for the Zika virus, rabies, cytomegalovirus, even some influenza. And so we've been doing animal studies and close to doing clinical trials for those vaccines. So the technology and the idea and the technology have already been in place. And that's why, with enough funding and enough people devoted to it, that's why we were able to make the COVID vaccine so fast. We were not really starting from scratch. Also, coronaviruses have been around for a long time, and we've known how severe the infections can be. The current pandemic is called, the SARS COVID virus, because it's related. It's very, very closely related to SARS. It has a lot of the same genes. So a lot of research has already gone into coronaviruses and mRNA vaccines. So it's no surprise that we were able to put all that together and actually make this vaccine so quickly. Again, tons of money, tons of people put to work on this. So regularly, vaccine development is slow because you'll only have 1 or 2 research groups working on it. and, so it's going to be slower. All right. There's no risk of infection. You only have mRNA for particular antigen, and that's it. And it's the spike proteins of the virus. So no risk of infection. You're giving the mRNA to the cells where you received the shot, so the arm muscle cells. They will then produce, they'll take in the antigen, display it on their MHC1 markers, and that will give a full adaptive immune response. T and B cells are going to respond. You're going to get a memory response. So it's good, very effective. Okay, so you do not need to memorize this. I just wanted to show you the common or the recommended vaccines and at what age. So this, I couldn't even fit all of it on one screenshot. So if you're interested, here's a link, and you can see, from birth all the way through 18 years. They also have other chart for adults, how often you're supposed to receive booster shots, for example. So yeah, vaccines are one of the best inventions, completely safe, prevent disease. We're saving millions of lives and increasing the quality of life for billions of people. Huge deal. Nothing in medicine is as valuable as our vaccines. Terminology: serology, study of the reactions between antibodies and antigens. Antiserum is a generic term for serum that has antibody in it. Globulins, we already talked about. They're serum proteins, they're globular, and if they're immunoglobulins, then they're specifically antibodies. So the type of the type of globulin that would be an immunoglobulin is called gamma globulin. And you might be wondering, why so many different names? Well, if you take the serum and you separate it on a gel, doing gel electrophoresis, you'll get these different bands, which means you have different size and different mass of particles. Each band, everything's identical, okay? And in this band here that I highlighted for you, this is your gamma globulins, and that will be all of the antibodies that you have, okay? And then these bands will be a different type of globulin called beta and a different type called alpha. They have different functions. And then lastly, we have our albumin. Remember, albumin is a molecule that is a carrier of things in your blood. So super important. And you've probably talked a lot about albumin in your physiology class. I don't think we talked about it much in our micro class. Okay, so if you forgot, or maybe you never learned what gel electrophoresis is, watch these two videos and it will show you exactly what it is, what you need to know. All right, so we can utilize this knowledge about antibodies and antigens to screen patients to see if they have infections, to see if they have immunity. We can also utilize it in research that we're doing that's not diagnostic. So diagnostic and serological tests. You can use monoclonal antibodies for all sorts of different tests. And you could do agglutination reactions, neutralization reactions, ELISA, Western blot, and you can even make antibodies that are conjugated or tagged with a fluorescent molecule. That allows you to do some really cool imaging. Unfortunately, we don't have time to discuss that, but we will focus on agglutination, ELISA, and Western blot. So a slide agglutination test is where you take these tiny beads. You order this from a manufacturer, and they're coated in a particular antibody for a particular disease. And then you would place these, you would have your positive control where you have the beads and the antibodies with the antigen. So you know what the reaction looks like. And then you would have a negative control, of course, which would just be water or buffer with the beads. And then, of course, you'd have your actual sample that you're trying to test. Okay, a positive reaction looks like this. It's going to look grainy because you have the areas where you got the clumping, you got the agglutination, okay? Whereas a negative is going to just look homogeneous, like nothing has happened, okay? We don't have the third one shown here, which would be the actual test sample. Just the positive and negative control are showed here, okay? This is a really neat short video that shows the test animation of what's happening at the molecular level. We do slide agglutination tests commonly against antigens like capsules, flagella, and LPS. So big repetitive antigens, right? And mostly we can use this for diagnosing UTIs or meningitis. So it is still used. As simple of a test as this seems, it can be very useful. And of course, you can apply this however you wanted to in research, right? So just because I'm telling you what the most commonly used forms are, doesn't mean that you can only ever do that, right? Once you learn about different tools, resources, and assays, then you as a scientist get to be as creative as you want to utilize all these different tools. Okay, so for slide agglutination test, I already told you you're going to add the antibody bead solution to each of the 3 areas on the slide. Then a few drops of the unknown sample will go onto one of those areas. A few drops of the antigen, which would be for your positive control, and then a few drops of water or buffer for your negative control. So all 3 will have the antibody, but only the ones that you give the antigen to will be positive. Of course, you don't know, your unknown sample could be positive or negative. And again, you always want to have a negative and positive control. 1, to make sure your supplies are good, your test is valid, and 2, so you know how to interpret your results. What does a positive look like? What does a negative look like? All right. ELISA is a bit more complicated of a test. You may have already done the lab this week where you did ELISA. If not, then look forward to it. Enzyme-linked immunosorbent assay. ELISA stands for enzyme-linked immunosorbent assay. You're going to test patient serum to see if they have antibodies, or you're going to test the patient's serum to see if they have antigens. So you can do this test, a couple different ways. In your simulation, you will be doing the indirect, where you're looking for antibodies, which means you have the antigen and you want to see if you can catch any antibodies in the sample. And that's good. That's perfectly valid. Sometimes you want to be able to detect a pathogen directly, and you would test the patient's serum for antigen, which means you would have the antibodies and you want to see if you can catch antigen. So you just need to know which way you're doing. The most common technique for both direct and indirect is called sandwich. This is where you use a secondary antibody that is attached to an enzyme. And then when you add the substrate for that enzyme, you'll get a color change. Looks like my N fell off. So the enzyme substrate will react when the secondary antibody is bound to the target. And this will make more sense when you see the steps. But after every time you add something, whether you're adding your primary antibody or your secondary antibody or the patient's serum, you do a rinse step between every step. So only things that are bound will remain in your test. Everything else gets washed away. So let's look at that. So direct ELISA, the goal is to see if there's a pathogen in your patient's serum. The very first thing you have to do is add this primary antibody. So you're going to start with an empty well, and you're going to coat it with antibodies. So let's try to draw that. So you would start with an empty well. Okay, so there's my well, and you're going to coat the whole well with antibodies. And so you can imagine when this is 3D, you would have a lot more than the antibodies I'm showing here, right? But hopefully you get the idea. You're coating the whole lining with antibodies. They're all bound. Their constant region is bound to the well. And then you're going to rinse. See, rinse. Okay. Then you're going to add the patient's serum. And if the pathogen is in the serum, it's going to get caught by the antibody. And we'll just say it looks like that. Okay. You can see it nicer here. And then you're going to rinse. So anything left over gets washed away. And then you're going to add your secondary antibody. And that's the real power here. And this is why it's called sandwich, because the secondary antibody has a bunch of binding sites on it. Right, see this? So just one secondary antibody can bind several substrates. And furthermore, you could have another antibody here. You could have another antibody here. And so, and each of those can bind several substrates, right? And so this gives you what we call amplification effect. You're amplifying the reaction. You're going to have a much more reactions taking place. Okay, and of course, then you rinse. And then you add the substrate, which are the boxes. And each of those boxes will then go into an active site on the antibody enzyme and get produce this color. Okay, so again, you have a rinse between each step. So if it's bound, it stays. If it's not, it gets rinsed away. So if the patient doesn't have the microbe, then your secondary antibody won't have anything to bind to, and it'll get washed away, and you'll have nothing happen, right? No color change. All right. So I'm not going to talk about indirect here because you saw it in lab. So direct sandwich ELISA is what we just went over, and I typed it all out for you. So make sure you read this and you understand it. Test yourself. Draw it out, write it out from memory. Make sure you understand the difference between direct and indirect. Here is also from lab an example of indirect. So now you're looking for antibodies, and this shows if it's an IgG antibody, right, it's a monomer. If it's an IgM antibody, it's a pentamer. And so we saw this in lab, and there's even an animation for you if you want to see it step by step. Okay, Western blot is the last test that we'll look at, and it's perhaps the most complicated. It's commonly used to confirm HIV or Lyme disease infections. This can also be direct or indirect, whether you look for antibodies or antigen. Whenever you look for antigen, it's direct. Whenever you look for antibodies, it's indirect, okay? So in general, you're going to run a gel electrophoresis, to separate proteins, which would be the antigens, okay? And then you blot those onto what we call a filter paper. And now that filter paper has the antigens. And then you wash the patient's serum. And if the patient has antibodies, they're going to bind to those antigens in the filter paper. And then you would rinse it with the enzyme-linked antibody, so the secondary antibody. And it will bind only if the patient was positive. And then you would add the substrate for the color change. And in this case, you would be looking for the formation of bands. So here again, overview of the process: gel electrophoresis to separate the specific antigens, blot it onto the filter paper, rinse it with the patient's serum, add the secondary antibody, add the substrate. If the patient is positive, you will see the formation of bands. If the patient is negative, then you won't see anything at all, except for your positive control, of course. Remember, whenever you're looking for antibodies in a patient, make sure you've given them 7 to 10 days post-exposure to have time to make those antibodies. If you're looking for antibodies in the patient, it's indirect. If you're looking for antigen in the patient, it would be direct. So then you would, if you're looking for antigen in the patient, then you would need to start your gel by having the antibodies, different antibodies. So again, depends on whether you're doing direct or indirect. Okay, and this is another image, another way to think of this amplification effect, right? So you have your primary antibody that has bound antigen. So in this test, you started with antigen, you found the patient has the antibody, and then you add the secondary antibody, which has the reporter or the color, right? And you can get several of those secondary antibodies for each individual primary. So you get that amplification effect, and it'll make it easier to detect, see a positive. Okay, now we have lots of test your understanding questions. Make sure you take time to answer them fully on your own before we go over them in office hours. And we're going to wrap up our studies on immunology by thinking about tumors and allergies. Okay, so tumors, you know, you have cytotoxic T cells and you have natural killer cells, and these both can kill cancerous cells. You know that there's lots of different things that can cause cell damage that results in the formation of a tumor, and that would be called a carcinogen. So if chemical can cause damage leading to uncontrolled cell division, it's a carcinogen. Benign tumors, they grow very slowly and they stay in place and they're not a risk to you. Malignant tumors, on the other hand, grow very fast and have the ability to spread throughout the body. And that's where you have to be very concerned and you have to receive treatment because that is when it's cancerous, right? And can lead to death. Okay. Sorry, guys, my voice is running out. Okay. Treatments of cancer. One popular treatment that will usually be done with a combination of treatments is using monoclonal antibodies that we already know the receptors on that particular tumor, and we can give antibodies against it, and that helps the immune system to fight. Of course, we also have other chemotherapy, chemical therapies, and we have radiation therapy, and right, all sorts of different things that we can do. But the use of monoclonal antibodies is really great because it doesn't have side effects like radiation and chemo. Immune system malfunctions when it attacks itself. You have autoimmune diseases. I talked briefly about diabetes type 1. If you're interested in learning about autoimmune disease, look at chapter 19. Your textbook talks about some of the more common autoimmune diseases. Hypersensitivity, or what we call allergies, happens. It can happen in actually two ways: immediate or delayed. In either case, the body has mistaken something that is inert, such as pollen, certain food molecules, dander, dust, mold, anything, practically anything, but your body has mistaken it as being a pathogen and has mounted an immune response against it. Typically, this results in IgE antibodies and lots of histamine being released. Okay, so two main types: immediate hypersensitivity reaction. This is what we're used to when we think of like sneezies, sneezing, itchy, watery eyes. That's the IgE antibodies and getting lots of histamine released in your mucosa. So mast cells release a bunch of histamine, and that causes some inflammation, itchy, runny nose, for example. But one of the dangers is that it can cause your smooth muscle to constrict your airways. And of course, that can lead to anaphylactic shock and death. So certain hypersensitivity reactions are potentially lethal. And that's when someone would carry an EpiPen, right? So if you are severely allergic to bee stings or shellfish, right, there's certain peanut allergies, right? Certain people have to carry those EpiPens around because they have an immediate hypersensitivity that leads to anaphylactic shock. But plenty of us have hypersensitivity reactions that are really just upper respiratory, nose, eyes, right? And then you just take something like Claritin. Okay, type 4, that is weird. Wrote this weird. This should be a Roman numeral. This should say 4. So Roman numeral 4, delayed hypersensitivity reaction. This is your poison oak, poison ivy, or our TB test. These we're going to see rashes on the skin, right? And this is due to cell-mediated branch. So no histamine. You take histamine, it's not going to do anything for you. Okay. Why? Cell-mediated, you would need to be able to target the T cells that are causing the tissue damage. So they're releasing cytokines, they're activating macrophages and neutrophils, and they're destroying some of the tissue in that area. And so usually it takes a day to a couple of days for the rash to even occur. So you go on a hike and the next day you have a terrible rash. You probably contacted some poison oak or poison ivy, and you would need to treat this with steroid. Antihistamines will not work for delayed hypersensitivity. So this is the immediate hypersensitivity where you get the IgE antibodies, and that triggers the mast cells to release histamine. And this is the delayed hypersensitivity, such as our TB test, where the allergen, in this case, the tuberculin molecule, causes this cell-mediated response that will lead to macrophage recruitment and giving a pustule or rash. So this is, if it was poison ivy, again, a delayed hypersensitivity reaction. And there's a couple different pathways that can take place. This first pathway leading to this tissue damage through macrophages causing tissue damage. There's another pathway that can happen where you have a reaction in here that, you know, you get the recruitment of neutrophils, which essentially is going to end up with the same result. Okay, so I think I included, did I? A short? Nope, that's it. Okay. Oh, do not bother memorizing this pathway. I'm not talking to you about all these specific things here. What you need to know is this right here. These images are just to help you remember this information here. Okay. All right, blood types, we've gone over this in lab, but just to remind you, there's A, B, and O, and each one could be plus or minus. And so there are antigens. Some people have the A antigen on their blood cells, on the surface of their blood cells. Some will have B, some will have both, some will have neither. And then there's a third type called RH, which we refer to as being positive or negative. So it's all talking about antigens, but we don't usually call them antigens when we're talking about our own cells, but because we've learned that there are only a few types for red blood cells, we do talk about them as being antigens. Okay. So the receptors on our red blood cells determine our blood type. Antibodies will be made if any other type enters the body, and that could be life-threatening because you'll clump up all the red blood cells that don't match your blood cells, and that can cause a clot or a clump to block a blood vessel, and that can cause stroke, cause heart attack, right? And so it can be lethal. So again, you could be blood type A. You have the antigen A on your surface of your blood cells. So your body will never make antibodies against this receptor, right? But because you don't have the B antigen, you can produce anti-B antibodies if you were ever to become exposed to it. You don't automatically have these antibodies, only once you get exposed. Okay, same thing for B. If you're blood type B, you have the B antigen. You have the ability to make anti-A antibodies if you ever get exposed to that blood type. And then, of course, people could be AB, where they have both. And in that case, they're not going to make antibodies of either, because that would attack their own cells. And lastly, you could be type O, where you don't have A or B. And in that case, you're able to make both antibodies. What was left out of this system is the plus minus. So plus minus refers to this additional antigen, antigen D. It's called the rhesus factor. And a person can have it regardless of being A, B, or O, right? And that's why people say I'm A positive, I'm A negative, I'm O positive, I'm O negative, right? That's the rhesus factor is the positive negative. If you have antigen D, if you have the rhesus factor, you are blood type such and such positive. Okay, if you don't have it, you're such and such negative. So this is particularly important when we look at blood transfusions and when we look at pregnancy. So if a mother is Rh minus, but the dad is Rh positive, there's a chance that the baby can be Rh positive. So what happens the first time an Rh mother is pregnant with an Rh positive fetus, during birth, the mother gets exposed to this blood type. So this baby is fine and healthy, but during the birthing process, during delivery, the mother gets exposed to this baby's blood from the placenta, and she'll make some antibodies. So if she ever becomes pregnant again with an Rh positive baby, those antibodies will cross the placenta and attack the baby, and typically will cause death of the baby. Okay. Then here we show a mother who's Rh minus, a baby who's Rh positive, but this mother has been given a special therapy called anti-Rh treatment. Okay. And so basically we get rid of the antibodies that the mom has so that the baby will be safe. So testing should be done every time a woman is pregnant to see if this is the situation. And then she'll be given this therapy. So that's hemolytic disease of the newborn, is when the mother has antibodies that attack the baby because she's Rh minus and the baby is Rh positive, but only subsequent, right? It doesn't affect the first baby that's Rh positive. And it has no effect on Rh negative baby, right? But second and subsequent Rh positive fetuses can be harmed by those antibodies that the mom has. Okay, now blood transfusions. These are harmful because you will make antibodies and they will agglutinate those blood cells. And so blood type O minus does not have any surface antigens, not A, not B, not Rh, 0. So they are the universal donor. Everybody can safely receive O minus, O negative. Okay. On the other hand, AB positive people can receive any blood type safely. So that's cool. Everything else you'll need to think about it, and that will be on your test. Okay, that is it. I know this was a very long lecture. Make sure you review all the parts. Make sure you watch your lab video carefully as well. They go together. All right, take care. Bye.